everybody. My name is Ella. I'm a PhD candidate in the Genetics, Genomics, and Bioinformatics program, and I work under Dr. Philippe Rolf-Salson. I originally came from New Hampshire, the University of New Hampshire, studying medical microbiology, but I have since switched my research focus into sustainable agriculture. I study grapevines. So grapevines are plagued by a disease called Pierce's disease, which is caused by a bacteria, Xylella fastidiosa. This pathogen can render the grapevines to have scorched leaves, raisening of the berries, and can eventually lead to the death of the vine. And so my project has two aims. The first is to look at the microbes, all the organisms living inside the grapevine, to see how the plant, the pathogen, and the microorganisms are interacting with each other. So the first part is to identify who is living in the grapevine, and the second part is to find some specific microbes as a biocontrol against our pathogen, Xylella, to figure out if there are certain microbes we can use as probiotics or biocontrols against the pathogen. So I'm in the GGB program here at UCR. I originally did medical microbiology at UNH, which is one of the great things about the GGB program is it's very flexible. Bioinformatics are necessary in almost all biology programs currently, so that's how in the program you can go to virtually any lab that studies biology, which is how I, as a medical microbiologist, switched to a plant pathology program in the botany department while doing GGB as my program. One of the other things I really love about UCR is my PI, Philippe. I've really gotten to known him over the years, and it's been enjoy working with him. He's very supportive, not only in his role as my mentor, but also in supporting me in my outside and outreach activities. While at UCR, I was able to develop 2 different graduate student programs. I was also able to study abroad for 2.5 months in China, as well as organize a number of outreach programs, both for sustainable agriculture and for sustainability at UCR. GGB stands for Genetics, Genomics, and Bioinformatics, which is kind of looking at the DNA structure of anything in the world and understanding, using computational analysis to understand at a deeper level how living organisms are functioning. In the GGB program, we have people who are currently serving under departments of microbiology, plant pathology, botany, neuroscience, biomed, basically any sorts of biology programs or departments a GGB student can find and join their lab and find some computational project to work on within the concepts of the PI's lab. Hi, it's Ella again. I'm here to show you one of the great assets we have here at UCR. It's our genomics core. So if you follow me, I'll show you around. We're here at the genomics core at UCR, which is equipped with all of your sequencing needs. This is a communal spot shared among students at UCR that is equipped with things like our sequencers, NextSeq and HiSeq, as well as MySeq. And you can do everything in this core from quantifying DNA to sequencing libraries. So I use this machine to understand what microbes are inside of the grapevine. I've used machines in here to also quantify all of my DNA and to quantify my pathogen that is in our grapevines, which we previously saw in the greenhouse. So I hope you enjoyed our tour of the genomics core, but I also just want to say that we have a number of other cores here at UCR, including our metabolics core, metabolomics core, our proteomics core, and our bioinformatics core. So whatever your needs are, UCR has a core and supporting staff to help you out. Well, that's it. I hope you enjoyed the tour of the Rolf-Salson lab and hearing about my research about grapevines and their pathogens. I hope you learned a little bit about GGB and the program and all that it has to offer. And don't forget to check us out on Twitter.